My name is Taieb. I am a nuclear physician and I'm working at Latimone University Hospital. And we are working with Dominique Barbolozzi on several models. And my main topics is on the management of paragangliomas and here. And we will discuss about the potential role of mathematics in understanding phenotypic differences across hereditary paraganglioma syndromes. So a background. So as you know, during embryology, there are some cells which are pluripotent, and these cells are destined to migrate and to differentiate it into various tissues, including some neurons, for example, sympathetic and parasympathetic neurons, but also some endocrine cells which are located in the adrenal glands, also called the adrenal medulla, and extra-adrenal paraganglia. For these paraganglia, some of these paraganglia are closely in relation with the parasympathetic nervous system, and the prime example of this paraganglia, which acts as chemoreceptors, is a carotid body. The carotid body is a small structure completely embedded in the wall of the carotid artery. And this structure is able to detect the concentration of oxygen within the blood. And when there is an hypoxemia, the carotid body is involved in a reflex that will act on respiratory muscle to induce hyperventilation in order to compensate, the hypoxemia and to restore oxygen concentration. So these structures are very, very important, and they are in close relationship with the autonomic nervous system. Some of these cells are located in the head and neck, and they are associated with the parasympathetic nervous system. And some of these cells are located in the abdomen, in the adrenal here, or, in the retroperitoneum, just near the aorta, and they also act, as chemoreceptor. Most of this cell will regress by autophagy during embryogenesis, and they probably act during pregnancy to monitor and to control blood pressure. So, these cells are widely distributed within the body, from the skull base to the pelvis. To summarize, we can say that there are head and neck paraganglia, which are in relationship with the parasympathetic nervous system, and abdominal paraganglia, including adrenal medulla, which are in relationship with the sympathetic nervous system. And in few cases, some tumor may arise from these cells, and they are called paragangliomas. For example, here it's a tympanic paraganglioma. These tumors are highly vascularized, as you can see here. It's an angio-MR study. You will see here. This is a tumor. Okay, and when the tumors are located in the adrenal medulla, they are called pheochromocytomas. So the whole family of this tumor is called paraganglioma and pheochromocytoma. Okay, and these tumors are highly vascularized. Here, it's an example of an angiography after embolization, and you will see all the vessels. So it's really very rare, but potentially devastating disorder, because this tumor can secrete adrenaline, noradrenaline, but also are very difficult to be removed with many postoperative morbidity. Okay, so the role of imaging is to detect this lesion, to potentially detect multifocality. Here you can see on PET imaging 3 different lesions in the same patient. One here, 2, and the last one here. But also potentially to detect distant metastases that could be everywhere in the body. In this patient, here it was a FIO, the large FIO, with many bone metastases. From a genetic standpoint, these tumors are very important, because more of 30% of these paraganglioma are familial. At least there is 20 pheoparaganglioma susceptibility genes, but mutation in the subunit B and D of the gene succinate dehydrogenase, SDH, B or D, are the leading cause of these familial tumors. What occurs in patients? Patient with SDHB or SDHG mutation, in this patient, all the cells in patient are mutated with a single gene mutation, which is inherited from the parents. But, you know, tumorigenesis will require at least a mutation, a second hit mutation, you know, in the second allele. So it's a B-allelic inactivation of the gene that will lead to tumorigenesis. So the SDH, it's a complex with 4 subunits, A, B, C, D. Mutation in B or D are the leading cause, but whatever the gene involved in the disease, you will have a complete loss of the protein. The substrate of the protein is succinate, so you will have a SDH deficiency and an accumulation of succinate, and this is clearly shown here with highly elevated value of succinate, and succinate acts as an oncometabolite and contributed to various pathways involved in this tumor, including the activation of the vascular growth and proliferation. So it's really interesting because you have 2 main familial disorders, patient with SDHB mutation, patient with SDHD mutation. The molecular consequences are the same, the biochemical signature is the same, but the phenotype are completely different. For SDHB patients, most of the tumor will be developed within the abdomen. But for SDHD patients, most of the tumor will be developed in the head and neck. And there are also many differences regarding the penetrance of the disease. Almost all patients with SDHD mutation with B-allelic mutation will develop the disease, but only 20 to 40 patients will develop the disease for SDHB. Most will have abdominal paraganglioma, some head and neck paraganglioma, but the coexistence of the 2 diseases, of the 2 locations in the same patient is very rare, less than 3%, and the malignancy is highly elevated, 30%. But for SDHB, it's completely different. High penetrance, most of the patients will have head and neck, very small cases with abdominal, very rare coexistence between the two kind of location, and malignancy is very, very rare. So what drives phenotypic differences between these two disorders? And phenotypic differences between SDHB and SDHD are currently unexplained by molecular genetics. So we have decided to study and to compare the disease dynamics of these two conditions via a Markov chain model based on 4 clinically defined steady states. So the model is here, 4 steady states: no disease, head and neck paraganglioma, sympathetic, which also means abdominal paraganglioma, including FEO, and metastasis. And in the model, there are some transitions, as you know, probability for here tumor formation, here tumor regressions, here probability for developments or regressions of metastases. Here's probability to stabilize in a clinically defined state. And here is a transition matrix. So we have made some assumptions. First of all, the transition probability here was fixed. To match the number of free parameters with the number of steady states, it was assumed that transition probability do not depend on tumor location. Okay, so this both probability was considered to be equal, as well as this one. And since, you know, the coexistence of both type of location, parasympathetic and sympathetic, in the same individuals is very rare in clinics, this probability was considered to be fixed, to be 0. So what's occurred in SDHB mutation carrier and what the model provided, it's very interesting to completely fit with all the observation from clinical data. You know, these patients are characterized by, you know, a very low probability to stay, you know, in these steady states, head and neck or sympathetic. But when tumor gives rise to metastasis, there is a very high probability to stay in this condition. And all the parameter completely fit, you know, with observational data. What occurs in SDHB mutation carriers, you know, all the patients will have tumor. The probability to stabilize, you know, in this situation is very high. But, you know, when tumor gives rise to metastasis, it's really rare event, but the probability to stabilize is very small. So, you know, here the estimated percentage at different clinical steady states in these two conditions, and this data are provided by the model and are in complete agreement with clinical observation. Here, 10% of all patients will develop metastases, which correspond to the 30% of patients with FEO and paraganglioma that will give metastasis. So this is completely, you know, the same. And this is also very important, as you can see here, the probability to maintain, you know, in a tumor steady state is very high for SDHD and very low for SDHB, but it's not the same for the probability to maintain in a metastatic steady state situation, which is very high for SDHB and very low for SDHD. So to summarize the finding, FEO and paraganglioma maintenance seems to be reduced compared to SDHD, due to the higher probability of tumor cell regression in B versus D. But when SDHB tumors give rise to metastatic cells are able to survive with decreased probability of regression compared to D. And for SDHD patients, they will all develop tumors that will be stabilized at this condition, at this state. However, compared to SDHB, maintenance of metastatic lesions seems to be less effective than for D. Now, when we move back to genetics, this finding potentially align with data suggesting that SDHD-related paraganglioma require less genetic events for tumor initiation and maintenance compared to B, and this pattern failed to initiate biology that promotes metastatic spread and metastatic cell survival in host tissue. By contrast, as we can see when we analyze genetics of SDHB tumor, there are a lot of mutations. These mutations are probably required to develop tumor, so the higher number of genetic abnormality is probably required for tumor initiation and maintenance, resulting in a lower penetrance of FIO and paraganglioma, but when cells give rise to metastases, they are assumed to be better adapted to sustain survival. So from this model, we could probably think about genotype-targeted approaches. For example, for the B patient, for the SDHB patient, since it's really easy to develop tumor for them, due probably to the ability and the very high frequency to have the second hit mutation, we should probably focus on prevention of mechanism involving tumor development and maintenance via prevention of second hit mutation via antioxidant or drugs that will reduce endogenous mutation or probably limit this patient to ionizing radiations. But for B, you know, tumorigenase is a less frequent event, but you know, patient will have probably small cell, you know, that can survive in host tissue. So maybe we should focalize our approach for killing cells at early stage of metastatic spread, either with no detected tumor by imaging, by, for example, adjuvant systemic therapies, or in patients with detectable tumor with radio or immunotherapy. So it's, you know, it could be different approaches for these patients. So in conclusion, the model shows differences in disease progressions dynamics between SDHB and SDHD disease, and this finding provide new cues for future exploration of B and D related paraganglioma behavior, offering consideration for future specific therapeutic or prevention strategies. Thank you.
Thank you very much, David. Any questions for David? It's a very complex field. I don't know how you manage to juggle between the D and the B. But I think it's also interesting because it's a field that maybe we won't be listening a lot for the moment, is about the what we call the pre-neoplastic. And so if I understood, the D is in a frame where regression is possible, and that of course immune intervention or other types of treatment. It's very interesting. Okay, so thank you very much. I think we can